Absorption refers to the process by which food components or their digested products, water, inorganic salts, and vitamins pass through the epithelial cells of the digestive tract mucosa into the blood and lymph. The primary site for nutrient absorption is the small intestine. As the main site for nutrient absorption, the small intestine possesses unique advantages. 1. Food is digested into small molecules suitable for absorption within the small intestine. 2. The small intestine has a large absorptive area. It is about 4 to 5 meters long. Its mucosa projects into the lumen, forming numerous circular folds. These folds are covered with a vast number of villi. The outer surface of each villus is a layer of columnar epithelial cells, and the apical membrane of each columnar epithelial cell has about 1,700 microvilli. These structures give the small intestinal mucosa a surface area of 200 to 250 square meters. 3. Food remains in the small intestine for a long time, approximately 3 to 8 hours. 4. The interior of the villi contains a rich network of capillaries, lymphatic lacteals, smooth muscle fibers, and nerve fibers. Nutrients can enter the blood or lymph via 2 pathways. One is the transcellular pathway, where substances enter the cell through the apical membrane of the villus columnar epithelial cells, and then exit into the blood or lymph through the basolateral membrane. The other is the paracellular pathway, where substances pass through the tight junctions between adjacent epithelial cells into the intercellular space, and then move into the blood or lymph. The mechanisms by which nutrients cross the plasma membrane include passive transport, active transport, and pinocytosis, etc. For details on these transport mechanisms, refer to substance transport across the cell membrane. 1. Absorption of water. Water is absorbed primarily in the upper small intestine via diffusion. The osmotic gradient created by the active transport of various solutes, especially NaCl, is the main driving force for water absorption. Water can be absorbed simultaneously through both transcellular and paracellular pathways. 2. Absorption of inorganic salts. 1. Absorption of sodium. Sodium absorption in the small intestine is active. The activity of the sodium pump, Na/Kat passe, on the basolateral membrane actively transports Na out of the cell into the blood, maintaining a low intracellular Na concentration. Na from the intestinal lumen enters the cell via facilitated diffusion, aided by carriers on the brush border membrane. Sodium absorption provides the driving force for the absorption of monosaccharides and amino acids. Additionally, a portion of sodium is absorbed concurrently with chloride ions. 2. Absorption of iron. Iron is absorbed mainly in the duodenum and jejunum. Iron absorption is closely related to the body's need for iron and other factors. Vitamin C reduces Fe3 to Fe2, and the acidic environment it helps create also keeps iron soluble. Both actions promote iron absorption. 3. Absorption of calcium. The primary site for calcium absorption is the duodenum. Calcium salts can only be absorbed when in an aqueous solution. Factors affecting calcium absorption include the body's demand for calcium. Vitamin D promotes calcium absorption in the small intestine. The acidity within the intestinal lumen. Bile acids promote calcium absorption. Phosphates hinder calcium absorption. 4. Absorption of anions. The electrical potential difference generated by the active transport of Na from the intestinal lumen promotes the absorption of anions. 3. Absorption of sugars. The small intestinal mucosa typically absorbs only monosaccharides, with small amounts of disaccharides. The absorption rates of different monosaccharides vary significantly. Galactose and glucose are absorbed the fastest, fructose next, and mannose the slowest. The absorption of most monosaccharides is an active process, occurring against a concentration gradient. As Na moves passively into the epithelial cell down its concentration gradient, It drives the secondary active transport of glucose into the cell against its concentration gradient. Monosaccharides that enter the cell via this process then leave the cell into the interstitial fluid via carrier-mediated facilitated diffusion and subsequently enter the blood. 4. Absorption of proteins. Amino acid absorption is similar to that of monosaccharides. The process by which amino acids enter the mucosal epithelial cells from the intestinal lumen is also secondary active transport. 3 main amino acid transport systems have been identified on the brush border of small intestinal mucosal cells, transporting neutral, acidic, or basic amino acids, respectively. Generally, neutral amino acids are transported faster than acidic or basic ones. Amino acids that enter the epithelial cells also exit into the interstitial fluid via carrier-mediated facilitated diffusion and are then utilized by the body via the blood. 5. Absorption of fats. After long-chain fatty acids and monoglycerides are absorbed, they are largely resynthesized into triglycerides in the endoplasmic reticulum of the intestinal epithelial cells. These triglycerides combine with the polypoproteins produced in the cell to form chylomicrons, which are then released into the intercellular fluid via exocytosis and subsequently diffuse into the lymphatic circulation. Medium and short-chain fatty acids and their monoglycerides are water-soluble and can enter the bloodstream directly via diffusion, primarily in the duodenum and jejunum.